Hi, it's Ms. Bastido here to talk about meiosis and sexual reproduction. Just going to kind of jump right in. We are going to start by talking about asexual reproduction. You might have learned about this, that most time it includes mitosis. And so you've learned about mitosis previously, but I just want to sort of revise it a little bit. So remember, in mitosis or in asexual reproduction, we're making identical copies. So we have a cell and we're making two identical copies of the original cell. And so we call that the parent cell, and we'll end up with identical daughter cells, or sometimes we'll call them clones. So you can see I've cloned my dog Diego here to help you remember that. And so we always start with a cell. That cell has to go through interphase, and an interphase is when DNA replicates itself. And so we did DNA replication in the last video. And so when DNA makes that identical copy with the chromosome, it's making that second arm of the chromosome. And so when the second arm of the chromosome is made, then it goes through the rest of mitosis, prophase, metaphase, anaphase, and telophase, and separates those sister chromatids into their own cells, and that leads to having the same number of chromosomes and the same genetic information in each cell. There are organisms, eukaryotes, that do asexual reproduction, such as yeast, some amoebas or parameciums, and even hydras can do asexual reproduction, but they're pretty simple organisms. And so when we talk about the more complex organisms, they're mostly doing sexual reproduction. And so if we were to do asexual reproduction, it just really wouldn't work. So if we started off, we have 46 chromosomes in every cell. And if we started off with an egg that had 46 and a sperm that had 46, then we'd end up with a zygote or a fertilized egg that ended up with 92 chromosomes. And so that obviously doesn't work. And so we have to go through a different process in order to half the number of chromosomes, so that when the egg and sperm meet, we end up with a full set of chromosomes, half from the mom and half from the dad. And so you might have seen one of these in another grade at some point. And so you can see here we have 46 chromosomes. This is called the picture is called a karyogram, or you might have heard it called a karyotype. And we have 46 chromosomes in your cells, 23 pairs. And so we say you have 23 pairs because if you look at chromosome 5 here, you have 2 chromosome 5s, one from your mom and one from your dad. Notice that these chromosomes don't look like the kind that you're normally seeing like cartoon drawings of, where they look like an X almost. So these are single-armed chromosomes, which means the DNA hasn't replicated yet. So this is a single-armed chromosome, but you have 2 of them, one from your mom and one from your dad. And so that would be true of every chromosome number that you have. So you have 22 pairs of what we call autosomes, which are the chromosomes that aren't sex chromosomes. And then you have a pair of sex chromosomes. So if you are assigned female at birth, you would have 2 X chromosomes. And one would be from your mom and one would be from your dad. If you were assigned male at birth, then you would have an X chromosome from your mom and a Y chromosome from your dad. These 2 chromosomes are actually really nothing alike, and the Y chromosome doesn't have that many genes on it that are important for life. But there is a gene on it called the SRY gene, which codes for the maleness, which is what kind of referred to as the maleness gene. On the X chromosome, there are genes that have nothing to do with being male or female. So we'll talk about those later a little bit. One thing to just notice is that you have 2 for every chromosome. You have 2, one from your mom and one from your dad. And that's going to be important here when we look at what's happening to the chromosomes during meiosis. And so we call those chromosomes, let's say chromosome 1, you have 2 of them, one from your mom, one from your dad. We call those homologous chromosomes. And that's because they have the same genes on them. And so I kind of put a yellow and a blue gene here, but they have the same genes on them. They just don't necessarily code for the same thing. And so while let's say there's a gene on this chromosome here for eye color, and there's a gene here for eye color. One could code for one color of eye color and one could code for a different one, or they could code for the same one. And so that's how those two chromosomes are different, but the order of the genes for the traits are the same on these homologous chromosomes. And so homologous chromosomes are going to be really important as we go through the rest of these slides. So if you look here, we have a single-stranded, I like to call it a one-armed chromosome, and then sometimes you end up with a two-armed chromosome. And so same genetic information on one arm as on the same arm. So we'd say these are sister chromatids. And so how do we make sperm and egg? Well, the process by which we do that is we have to reduce the number of chromosomes. So we start with a cell that has 46 chromosomes, and that through the process of meiosis, we will reduce the number of chromosomes to 23. And then the egg and sperm, which are called gametes, will meet through fertilization and will form a zygote, which then brings the number back up to 46. And so you can kind of see that here. We have to reduce the number of chromosomes. So we say we have to go from 2n, meaning two sets of chromosomes, to n, which is a single set of chromosomes, to get to a sperm or an egg. And so we say when you have two sets of chromosomes, it's called diploid. And when you have a single set, it's called haploid. And so sperm and egg in humans would be the only cells that would be haploid. All of our other cells would be diploid. And then when those two haploid cells, egg and sperm, come together through fertilization, the diploid number is restored. And so we have this production of gametes, which reduces the number of chromosomes, and then we restore that through fertilization back to 46. And you can kind of see that here. So we start with an egg and a sperm. Those egg and sperm come together through fertilization. We end up with two copies of every chromosome, and then that individual will then go through meiosis and produce their own, in this case, sperm, which would then go back to haploid. And so this is a cycle that happens, and all organisms do some kind of cycle that's similar to that. And so here is an overview of meiosis. We're not going to spend too much time here, but it might be a good picture for you to go back and try to maybe, if you could go, can you go through each picture and explain what's happening in each picture? Through overview of meiosis is that interphase, where DNA is replicated, occurs at the very beginning, and it won't occur again. So just remember, DNA is replicated once at the very beginning. Then we go through prophase 1, metaphase 1, anaphase 1, and telophase 1. That's meiosis 1. And then we go through meiosis 2, which is prophase 2, metaphase 2, anaphase 2, and telophase 2. So we're going to take a closer look at some of these as we go through. So in our first step of meiosis, DNA has to be duplicated. We have these one-armed chromosomes. We need to unravel that DNA, and DNA polymerase will make a copy of the DNA, and then we end up with the second arm of the chromosome. And so it looks like this, like we were talking about earlier. And so here I have 6 chromosomes. Here I have 6 chromosomes. They're just two-armed chromosomes. This happens at the beginning of mitosis as well, so this looks a little bit similar to that. But from here on out in meiosis 1, it's going to look different. And so in meiosis 1, we'd say that the main thing that happens is there's a division of these homologous pairs. So the homologous pairs will pair up in prophase 1, and you see that here in this photo. And I want to show you what that looks like. So if we go here, you can see that when these homologous chromosomes pair up, so the place where they're pairing up, where they're we call this a crossover, is called synapsis. And when they're all together, we call them a tetrad. Tetra, the prefix means 4, and so it's because there's 4 chromatids here, that's where that name comes from. But what you really need to know is that crossing over occurs. And crossing over is when pieces of chromosomes from the mom and the dad chromosome break off and exchange pieces. So this is what the outcome would look like. You'd end up with, this is, let's say, your chromosome from your mom, but it has little bits from your dad on there as well. And this helps increase genetic variation, which we'll get to later. After that happens, go back to this picture, after that crossover event happens, we go to metaphase 1, where the chromosomes line up in 2 lines, so homologous pairs, we'd say, line up at the equator of the cell. And then these homologous chromosomes will be pulled apart. So this one's going to move in this direction, and this one's going to move in this direction. At this point, we started here with 4 chromosomes, single-stranded, and then we end up with 2 chromosomes in each cell at the end of meiosis 1. So we have reduced the number of chromosomes by the end of meiosis 1. In meiosis 2, it looks a bit different. And so in meiosis 2, really it looks identical to mitosis. So if you know mitosis, you know meiosis 2. But you have 2 cells going through it. So I have 2 cells going through prophase 2, metaphase 2, they line up at the equator, but there's no longer homologous pairs, so they get in a single line at the equator, and then these sister chromatids get pulled apart to their own cells. And so you can see here now, I have 1, 2, 3, 4 cells that all have 2 chromosomes in them. We started with 4 chromosomes, we end up with 2 chromosomes. And in humans, we would say we start with 46 and we end up with 23. Notice they are single-armed chromosomes, but you only need 1 arm to the chromosome. You don't really need that second arm. It's just for replication. And so here's all the steps. When we talk about interphase, prophase 1, metaphase 1, the big important steps are prophase 1, metaphase 1, anaphase 1, and then understanding that in meiosis 2, we're just dividing sister chromatids like we did in mitosis. And so the reduction division is in meiosis, so we go from diploid to haploid, so it's 46 chromosomes to 23 chromosomes if you're a human. And then in meiosis 2, we're just pulling apart those 2 armed chromosomes, and looks just like mitosis. So then we end up with 4 cells at the end, each with 1 armed chromosomes, but they're all haploid. So they have half the number of chromosomes. So when we compare the two, we both start with a parent cell. We both start, mitosis and meiosis both start with diploid cells. So DNA is replicated. We have these 2 armed chromosomes. But then something very different happens in meiosis with the homologous chromosomes pairing up and them exchanging pieces, them getting in 2 lines at the equator in meiosis 1, and then separating into 2 daughter cells that are haploid. At the end of mitosis, we have daughter cells that are genetically identical. That's really important, and that they have the same number of chromosomes. In meiosis, we're making genetically different cells that are all half the number of chromosomes that you started with. And so what does this look like? Well, if we're putting it together here, we would say that we start with these cells, they undergo meiosis, we end up with half the number of chromosomes in sperm and egg. Sperm and egg then unite through fertilization to form a zygote. That zygote then goes through mitosis, through development to form a fetus, which will then implant into the uterus. So what is the value? Why are we doing all of this? Well, the value here is that independent assortment, there's 2 things. So we have crossing over. That mixing of homologous chromosomes increases genetic variation. When we do this switching in prophase 1, increases genetic variation. But there was somewhere else that we were increasing genetic variation, which is you have 23 pairs of chromosomes, so 23 homologous chromosomes, and the way in which they line up. And so the order that they line up in. They get in 2 lines, you can see here at the bottom, but how they get in that line is different every time. And so because of that, they could be all the dad chromosomes on one side and the mom chromosomes on the other, but maybe the first 2 are this way and the last 2 are switched, or you get the idea here. So you can see that when we talk about what are the number of possibilities of the way that they could align, because we have 23 pairs of chromosomes, it's 2 raised to the 23rd, which is over 8 million combinations. That's a lot. And so that is what makes you more just slightly genetically different from your siblings. And when crossing over is added into that, that really increases genetic variability to almost an infinite number of possibilities. Then lastly, we have random fertilization. So which egg and which sperm are fertilizing each other is random, and that also increases genetic variation. And so why is genetic variation important? We would say because it's driving evolution and providing variation for natural selection, which is a requirement for natural selection. We'll talk about that a bit later, but just kind of keep that in your pocket when we talk about variation in natural selection and where does that really come from. And so any two parents can produce a zygote with over 70 trillion possible diploid combinations, which really shows you how siblings might look alike, but they also can have a lot of differences, as you can see in these pictures. Now, the way that meiosis occurs in males and females is a bit different. So the formation of sperm is called spermatogenesis, and it occurs in the testes. And so in the testes, you can see we have this germ cell that is diploid. So this is one with 46 chromosomes. And then those will then eventually go through meiosis 1 to become secondary spermatocytes. Those secondary spermatocytes go through meiosis 2 to produce spermatozoa. And so these spermatozoa are here, and they'll go on to the epididymis, which is next to the testes here, and they will go on to mature and become sperms, mature sperm. And this process is happening throughout a male's lifetime after puberty, and they're constantly producing sperm throughout their life after they start in puberty. A female is a bit more complicated than that. And so females, when they have matured, so at puberty, all of their potential, what we'd say ovums, or if you want to call them eggs, those potential ones are all sort of in prophase 1. And so during maturation, meiosis 1 will be completed. So it'll complete, in other words, at the time of puberty, at the time of birth, all of your eggs, potential eggs or ovums, are in prophase 1. And then once you reach puberty, these cells will then go through meiosis 1, one at a time. And when they do that, there's an unequal division of cytoplasm at the end of meiosis 1. And so you end up with one, we call it secondary oocyte. That secondary oocyte has lots and lots of cytoplasm. The other one doesn't, and it will eventually just sort of disintegrate and get broken apart. It's called a polar body. So this is not going to be able to be fertilized. Then at some point, ovulation occurs. When ovulation occurs, the secondary oocyte is released, and it does not go through meiosis 2 until it's fertilized by a sperm. And so if you've never been had a fertilized sperm, had an ovum be fertilized by a sperm, then your potential eggs or your secondary oocytes have not gone through meiosis 2. So this is a bit confusing, but when it goes through meiosis 2, again, a polar body is produced. And so you have these polar bodies that are produced, they disintegrate. You end up with only one ovum, and it will only go through that meiosis 2 once it's triggered by fertilization. And here's another look at that. And so here you have the ovary, and so inside that ovary, the secondary oocyte's released. That's after meiosis 1. This happens once a month on average. And it comes during ovulation. It goes into the fallopian tube, or sometimes these are called oviducts. And the oviduct, if fertilized by the sperm, if the secondary oocyte, if fertilized by the sperm, will then go through meiosis 2. And then it will produce one ovum, and it already has the sperm DNA inside of it, and we won't really get into too much more about that. But then eventually that becomes a zygote when it's a big cell. The reason that we have this unequal division of the cytoplasm is because until the zygote develops and is able to implant into the uterus to then form a connection through the placenta with the mother, it has to provide its own nutrients, and the sperm is not providing any nutrients. It only provides DNA. And so the oocyte, or the ovum, has to have all of the cytoplasm to be able to do these cell divisions, mitosis, after fertilization until it implants into the uterus. Hopefully that helped clarify meiosis for you. I think probably one of the hardest parts is just understanding what's happening between males and females and how that looks different. If you have any questions, let me know.